Oh, gonna record it. Nice, fun. See, some good things came out of COVID, right? You guys can review and everything. Now, I'm not gonna speak off the slides. It's kind of rude. But main goal in the class is, this is a challenging class. You need to meet the outcomes. Every bio professor expects that I will send you off with a very decent understanding of genetics that you can apply to plants, medicine, animals, wherever you want. Now, the balance here is that, yeah, it's going to be challenging. Those outcomes are tough, but I'm here 100%, okay? That's why I love teaching this class, is because this is the one that made me want to teach, actually, when I took it. This was my favorite class I ever took when I went to school. He comes by. I did this last year in the same room, don't worry. So we are in a very unique position with education right now, right? My tests are open note. ChatGPT can get you guys a lot of answers. So I have to design things and make sure that you're building past that, right? That's why a lot of the learning styles will be different. Exams do take up a big chunk of points, but so do a lot of the psychomotor skills that you see in lab. A lot of thinking and like intrigue stuff that you will see in the small writings. Everybody has that individual talent. And ultimately, like I say on the bottom here, and I have redesigned later slides to fit that bottom issue we have, the goal is always that this class is the best one you ever take, and that's always the goal. I will fail that goal, I understand that, but that's okay. Setting out with it, that's the plan. All right, hooray. This always helps everybody. What's gonna be on the test? Well, if it's in red, like you can better well think about it being on the test. I'm not gonna ask you to raise your hand, but if you're colorblind, you've gotta come find me or email me, and I'll put a little asterisk next to my red slides, okay, so that you know that they're red. It's kind of funny, but it's not. It's okay, don't worry. It's a genetics class after all. So blue is always the one that's a little different. It's like, it's either too great a detail for me to ever need from you on a test, and it's like, that's kind of unfair, or it's just like a helpful thing you can apply to some of the red stuff. Green, I really do mean like, you can relax, it's a cool story, it's a cool application, you're gonna be all right. So, despite all this support, the office hours, let's do the reviews, let's do the group studying thing. I'm there all the time if you need me, okay? At the end of the day, you gotta know it to pass. That's the end. And that's where we want to get there. Now, this is what I always say. You failed me, genetics is hard. No, genetics is just hard. I'm not hard, I'm nice, okay? A coach can't win though. You've gotta win that game. I can't take that test for you. And like I said before, I can't schedule you guys up against a very easy team. It's just not the nature of this class, I'm sorry. But that's the plan. Okay, actually there's some addendums to this one, I like that. So basic class structure, I don't know how much we need to do here. I'm actually gonna leave these up for 7 days, an entire week. So if you have some emergency or you have a game, or performance, something's happening, you have it for a week. You've also got the key info. One adjustment I made with the key info, I actually include a lot more blue and green text. That was actually some feedback I got last year, like midway through, and I was like, yeah, I can include it. The key info PDFs don't fall into the trap of only taking the notes that I take on the test. Try and make those images your own, if that makes sense. Try and make them in your own study guides, things like that. You guys see the YouTubes of the pre-med guys going crazy, making these big, beautiful, and ladies making these big illustrations of the whole class. You wanna do that. Don't rely on my images, make them into your own, combine things to yours. Everything's free. I don't believe in the racket of textbooks. Genetics isn't owned by anybody, okay? Everything's good. Sometimes when I post a link to a video or something, it's typically not gonna be on the test, it's usually just fun. A lot of reading links, video links, and module links are on the syllabus. They're in that kind of green and gray stack that you've seen. So if you're saying, hey, what do I read? What do I want to get ahead on? Just go to the syllabus, you'll see that big stack and you'll have plenty of time to do that. So another unique element is I do try to incorporate writing in here. It's a good chance to get into some of the cooler stuff. I can't test you on everything on an exam that is actually good for emotional intelligence or would otherwise express those talents. Equally, we'll do a lot of practice questions in here a lot of the time. And that'll be a way to hey, get points, get revisions, etc. I'll provide those revisions. I will just knock you for a 0 if it's not there. Don't be sad, I promise. I don't think you're a 0 out of 6. It's just that if something's there that you need to fix, I'm gonna give it a revision. Then you can come back and actually make the change, right? How many of you gotten revisions on a paper and it's like, sorry, you got a 60, you suck. That's it. And I would always get that and be like, oh, chill, thanks. This is so fun. I'm learning so much. So if we give revision, if I give that revision, put it through. You would be surprised the amount of people, though, that would do the writings, that get a revision, and then it would just sit there stale forever. So don't enter that population, please. So equally, you just let me know if you need an extension on stuff. Like most of this is like, I need it just, having it physically exist is the more important thing than the accountability. The accountability does exist in that in this class. There will be times that you'll lose points if you just don't show for something or something like that. So part of this class is getting you up to speed on that level too. Okay, labs, hooray. I was told last year, even by a student in the reviews, and somehow the tenure committee was like, oh, there weren't enough labs. They got out too early. So guess what? Now there's more. Sorry. You'll learn more though, and things are a little more streamlined, and I cut out some of the ones that were a little like, a little boring. So there's 18 people in most lab sections. You can kind of do a lot of the stuff as groups of 2, honestly. That way you get your hands dirty, everything's good. Again, remember, these are the stuff I can't test on the exam. So when we do flies, for example, animal people, that's your first animal, like, organism we're gonna trust. We're not gonna trust you with a grizzly bear before we trust you with a fly, okay? Equally, pre-med people, the fly, it's a very good little patient. If it squishes, nobody's gonna sue you. It's your first patient, okay? It's your practice here. That's what the translation is that the ability to like have that psychomotor skill, that thinking, that design, that open-ended, it's cool. And I think it's one of my favorite things because a lot of these labs are open-ended, specifically mutagenesis, because that kind of opens the door for people that aren't exam people to succeed so high. And it's always like super cool. Don't miss the lab. I've chased down too many people. I'm tired. I'm old. I don't want to hunt anymore. Okay. So I added this slide too. It's black text is usually sort of get your attention, sort of just a statement. It's always tough to have the conversations at the end of the term, and I had them in immunology this year, where it's like, oh my God, I failed, it didn't go well. So for me, the beginning is the end is the beginning. These are the conversations I wish I would have had with people. I think this is posted, if I'm right. Is this posted? Yeah, nice, sweet. Okay. So when the day comes that we have that conversation at the end of the year, could you have checked off these boxes? Now, not everybody needs to go to the reviews. That's okay. That's not for everybody. Not everybody wants to study in groups. People suck. It's okay. These are suggestions, though, that you can take on. That big study guide tier list that I've given you, I can walk you through those, coach you, figure out a diagnosis, a plan, everything, okay? That usually doesn't start until after exam 1. That's why I incorporated quiz 1 last year, and that helped get people a little like, oh, okay, that's actually gonna happen. So if you're looking for things, yeah, this is a list, but I'd rather discuss things with you and say like, let's use your talents here, let's ditch that strategy, let's move here, and let's get every of your chess pieces on the board that make the biggest difference. Hooray, enough of me. Well, actually, wait, one more thing, 5 more things about me. All right, and then it's you. So I'm from Colorado originally, awesome. This is now very Instagrammable, Hanging Lake. We used to go up there and jump off that thing. It's highly illegal now, oops. Sorry. I went to Carleton down the road, very tiny, lots of Frisbee, overrated, it's fine. Okay, Omaha, actually underrated. Omaha Medical Center, hooray. That's where I did my PhD. It was super fun, actually, yeah. So if you guys ever want to talk about Omaha, I'm a big fan, actually, surprisingly. Hey, and it actually doesn't cost $3,000 for an apartment to rent in Omaha. Who would have thought? Speaking of $3,000 apartments, Mayo, oops, well, Mayo was a good time, learned a lot. Got back into Minnesota, and here we are today. So, fun times, brief intros, who you are, what you're up to, what you kind of hope for the class in the future. So go ahead and introduce yourselves to your neighbors a little bit. All right. So we'll go ahead and move on about 30 seconds. You're good. You guys are a lot more targeted than last year. COVID was still a thing last year. That's not fair to say. Nice, there it goes. All right, so a few more informal things. I might actually get started. All right, here's kind of a scale of how I am usually too. There's me in Phys Labs. Some of you have had me in Phys Labs, clearly. I'd say the stats in the bioinformatics class, it's kind of like, well, yeah. Genetics, yeah. I mean, like I said, you got to pass the class. Immunology, I think perhaps I treated you too harshly. Immunology. Last fall it was tough. Tried to make a 400 level class into more of like a pre-med class that I had seen and taken before, and I was like, oh, maybe we're not there yet. Oops. It's okay. That's why, again, you can always adjust the class how it's going. Now, I don't care if you've seen this before. This is awesome. Sweet. So this is like a National Geographic winning photo. Hooray. The genetics in this photo, as you can tell, very cool, scary. I think you know what's coming. Sweet. We made Pomeranians out of them. Turned wolves into this. Good dog. All right, and that's my Pomeranian rescue Pom Scout. She's very rambunctious and very bad, so you'll see a lot of pictures of her. So the cool thing, as funny as the photo is, genetics where it's going is that Scout's genome is closer to the wolf's genome, the raw code, the raw DNA, than the wolf's genome is to a coyote. Something to think about. It's not the recipe, it's how you use it. It's what genes turn on or off. It's what genes go high, lots of pieces of them, or low. So for example, a lot of growth genes in a Pomeranian, they're gonna go lower, for example. But that gene still exists in both of them. Fun word for that is epigenetics. We will get there, but for the moment, we'll still see we'll build up to it. But that is a goal of this class, is to get into that actual molecular, I don't want to say real, but it's very much where the mystery lies in a lot of humans, medicine, everything. There's Scout, and as a size comparison, our little rescue German Shepherd Cannon. He's very good. He rests a lot, but he's a good dog. Equally, what do? So essentially I do cancer research. I try to combine three things. I try to combine patient clinical outcome, how long did they survive, what was their age, et cetera, that. Try to combine that with genes and say, if you have a mutation in this gene, you're gonna die sooner, for example. And it's kind of grim, sorry, cancer is not a fun thing. But equally, how can we use these genes up and down to predict the course, maybe of a different drug, maybe of a different therapy? Now the goal would be someday that you take people in this red curve right here and you treat them with something different so that they don't suffer that same detrimental outlook. And what we call this is genomically targeted precision medicine. It's fun, right? Gets you a lot, gets people excited. But basically, can you use genetics to predict the course of disease before it happens? Absolutely. That's the plan. Things like, I don't know, all kinds of cool stuff is in the works here, and I'm hoping to give you that intro so that this becomes so accessible to you. And that's sort of one of the cool things about biology is you don't want to outsource your health and that of your family. You'll know more. Now, as far as genes go, this is always a fun quote. There's a lot of philosophical stuff about like who you are, what your genes are, what your blood is, right? You'll hear that phrase, especially in Game of Thrones. It's got good Valyrian blood, for example. Well, a little oversimplified. So, as a practice, who knows identical twins in their life? Yeah, you got a couple, right? They probably look pretty similar, I imagine. Sorry for the creepy example, but whatever. How are they different, though? How can two people with identical recipes for DNA, for genomes, be different? Oh yeah. So any of those traits, like personality, for example, they can shift, right? The environment can move things, it can trigger things, it can suppress things, right? So treat this as your first little practice writing, for example. I'm not gonna have you turn this one in. So you don't actually equally, don't have to write, but cognitively it's fun for you, trust me. So what are ways that things are not genetic? What are things that are malleable to genes? What are things that can change over time? To think to yourself or write it down. And if it helps, you can think of things that are 100% genetic. I'll use this example later, but your blood type, just a couple genes, that's it. There's no way out of it, right? There's certain things that are 100% genetic. What about language, though? Could you separate these two at birth? Teach one German, teach one French? Is there any difference in how they would learn that? No. Those are sort of the bumpers, 100% to 0% influence on the beginning. And in the middle is where all the mystery lies. And part of this class will show us where we can begin to sort of address that. So, let's talk a little bit about the beginning. Ex nihilo, Greek translation, basically something from nothing. This was the beginning of heredity. Nothing exists and then something exists. That's our best philosophical guess here. Sorry, couldn't have more. We moved on later to, it's just the dude, that's it. Yeah, sure. We thought that there were, this was closer. They thought they were these things, these pieces of humans called gemmules that would somehow influence the embryo, and thus the offspring. But it was all the dude, so very wrong still. They're doing their best. They don't have biology or microscopes at this point. Yeah. So we eventually got to something in red text here. So yeah, this is real. This is week one. Yeah, sorry. Because we have to start from the reasonable beginning to make it to the end. That initial assumption was just that all inheritance, everything about you, is just a kind of a blend. That's it. And this was so accepted that it was just, well, everybody's going to blend up and that's about it. Now, what about this, though, doesn't make too much sense? Think intuitively, right? You don't have to say anything. Oh yeah, so by the way, when I say like, who thinks this? I actually am just saying you can kind of think it. Don't worry, I'm not going to call on anybody yet. We'll see. But this was our initial model of heredity. It's just totally even. Everything is just complete hybrid. That's it. But you can probably think of examples where that didn't happen, right? Who's ever seen two really short parents with a gigantic kid, right? Yeah. It's okay. Or two really tall parents equally with a really short kid. So this is the best we could work with. That was our starting point, okay? And we're looking at a lot of, usually we're looking at pretty complex traits. So things way more than color, right? We also weren't really factoring in environment the way that plays yet. So, bigger issue began with Darwin and his explorations. His explorations, oddly enough, were funded by the British military to go and secure islands around South America. So It was sort of one of the journeys that took us here, strangely enough. But the question he asked was, why are they different? Why would these things have different beaks? Why wouldn't they just all blend together, right? That's the accepted thing. Because you look at this guy, that's a little more narrow. That one's a little sharper. See that curve right there? And then this one's huge to like crack seeds and stuff, right? Why would they be different if they're this close and they could just go between the islands? So, if given the opportunity, life, biology would create infinite populations. Babies would have babies exponentially. Unfortunately for everybody, well, everybody involved as far as the offspring, food runs out, resources run out, predators show up, populations never make it to infinity. Those that can find the resources and those that can survive whatever ailments may be around in their environment, predators and other things, disease, for example, they would be the ones to have babies again. And over time, mathematically, in the random pool of all this, if you're on an island where you eat insects, that's my attempt at like an ant, dang it. Sorry, okay. If you eat insects that are like inside a tree, or if you just have seeds to eat, you're gonna crack those open. In that fight for resources, when times would get tough, and that's an important thing about natural selection and that millions of years long process is that abnormalities would have the advantage suddenly when there's a drought. Abnormalities would have an advantage suddenly when there's an insect bloom, that they eat all the plants, but now there's tons of insects to eat, so who's the winner now, right? It's that randomness that drives this. One thing that's very tough to get across in biology, the population, the individual doesn't evolve, the population does. That is time. There was one thing, there's another guy that doesn't get as much credit here, Wallace. He collected so many samples from South America that could show that diversity and show that related things could be alike, but have unique characteristics built for their environment, webbed feet, long bills, things like that. Now, in both of their fight to say, okay, we think we have a model for heredity. First argument against them. There's no foundation for it. We don't know what heredity is. We don't know of a molecule. We don't know of a thing that passes from dad and mom to son and daughter. We have no idea what that is. So your theory can't be true. There's another problem, and it relates to blending. All offspring would just be this nasty gray zone. There would be no specificity. There would be no freaks. There would be no weird animals that would take that, suddenly that advantage. They would all just be the same. And the real thing here that's tough is that, well, because remember, and I should have premised this before, everybody believed in blending. That was it. You don't have Mendel yet here. That's the problem. You don't have any of that stuff you saw in 105 yet. So they're assuming blending is at the issue. So they're like, that's impossible. Everything should be the same. Sorry. Kind of touched on this too quick. But in that variation, and this is a clutch of mice from two distinct parents, totally possible. We'll look at melanin genes. We didn't have a chemical, we didn't have a thing, a substance that was heritable. We had no idea. So you got to remember this, like the stage they were working from with these assumptions, why it was so frustrating, why it was so tough to say like, hey, we think things have an advantage if they have a better feature. Where would these anomalies come from that would be the ones that would be the successors then? And he would get shouted down on a lot of his tours, him and Wallace, saying like, hey, we think that there's variation in these populations, and then where does it come from? It's random, it's stupid. You have no foundation for this. Last thing that happened, something unique. You guys have seen Lamarck before, that evolution where it's like the giraffe stretches its neck and it like lives longer because it can now stretch, hooray. Yeah, that doesn't really happen. But if you look at the Lamarck evolution, and that's why it's in blue here as a good example. I'm not really going to test you on Lamarckian evolution. It's a great example to play against regular heredity, though. So Lamarck was a French guy that was like, okay, well, they're wrong. It's not populations, it's individuals. So if the trees in the savanna are this height, here are giraffes right here, they keep getting longer, and it's the offspring that will sort of inherit some sort of like stretching neck that will like get them up there. That's not feasible, sadly. And this would happen over a few generations, and it would just be like, they'll just adapt to survive, that's it. We know that's not true with how many extinctions we've seen. Can't actually have that happen. So, got to remember with genes and Darwin and stuff, it's the population that has the variation. Somewhere in that giant population, there exist taller giraffes. They will suddenly have the advantage. And suddenly all their friends and cousins and everything will die. They'll be the only ones left to have the offspring, and now all offspring will reflect how they looked. Now, the interesting thing that soon enough in the class we'll be able to come back to, honestly, probably in the third phase of the class, the final phase. Can organisms activate genes and in situations to survive? Sort of like a last stand sort of thing. Since it is possible. Lamarckian evolution is extremely possible. But typically at the microorganism and disease level, we'll get there. So, fun times. They're trying to tour, they're trying to say like, hey, populations have variation and the best ones live. And they're like, nope, you guys are idiots. Darwin would spend his last like 20 years combing through every scientific journal. And remember, this isn't on the internet. This isn't even in accessible libraries. You got to go where the book is. So he'd look through science manuscripts. He would look and look to find anything for heritability, any sort of substance, any sort of pattern outside of blending, and it wasn't working. He missed one. And he had taken notes on the magazine, the journal itself, and he missed one paper. Kind of tough. And this had been done 40 years before Darwin. So going into the past, Gregor Mendel's a monk. He's pretty bored. He's hanging out. He has a fascination with variation and offspring and future and things like that. Now, the fun story here, we all know what's about to happen. He's going to put the plants together. This actually started, he had mice, actually. He was doing mouse experiments on it, on heredity, and he was figuring it out. He was figuring out dominant recessive. His boss, the main priest guy, was like, that's a little too close. Don't do that. Don't find the heritable material for a mammal, please. You can do your plants, though. That's fine. So he actually did get shot down. It's also a cool story that he thought differently. Darwin could try and reconstruct what had happened. Mendel was very good at looking at like the micro level and between the small organisms, not a population, but not a large population like Darwin, but individuals, parents, offspring. So, oh yeah, well, Mendel's also kind of weird. He would talk to the pea plants too. He's kind of a weird dude. But hey, wouldn't you be if you were stuck in a mountain in Germany and like nobody talked? Be kind of tough. Okay, so he started with one that was a very good reputation of blending. Some of the pea plants are tall, some of them are short. And this was a very defined phenotype. There's our first word, hooray. You guys know that one, don't worry. Just gonna put it on there for us. Okay. So he takes a couple stocks, a couple other good words in here. What does true breeding mean? That means that when you breed a tall with a tall, it's gonna make another tall plant over and over and over again, hundreds of times. True breeding. Every time, that's gonna be how it goes. Same thing with this little short stock that he had. Could breed the shorts with the shorts over and over. Always the same height. And he noticed it was a pretty defined height. Given this, he thought, if this was blending, this should be a clear answer when I put these two together, right? It's the clearest of his phenotypes, it's the clearest answer that it was. It's like, hey, if this is blending, we know exactly what's about to happen. We're going to get a medium. Wait, it is. Okay, wait, yeah, it totally is break time. Okay, so I'll give you the time to think about that. What is he going to get when he opens it up? But I've also been talking for a long time. So, and this is a long class. I'll typically always try to make time for breaks where you just kind of like, you can get your phone out, you can hang out, you can either check through your notes, anything you want, you can come up, ask questions, things like that. Totally okay. I'll typically throw a picture of the dogs up. So, cool times. I know this is and we'll finish our story in a sec, because I know this is a long class. Oh, and don't worry, nobody's actually secretly Zooming. Those three people are the room, my computer, and the iPad, okay? So there's no way you can Zoom into class. How dare you even think? All right. Yeah, the breaks are like 2 minutes. I always feel kind of bad because it's like just not enough time to use the bathroom. Sorry. All right. Equally though, I'll just bore you with some old internet history. Look at this. Do you guys ever remember this channel? That old dude that blends stuff? It's super weird. I don't like it. Like he blended marbles once and it was like, don't try this at home. The glass dust will kill you painfully. And it was like, oh, why did we do it then? It's a good blender though. Oh no, I gave the answer away. But okay. I mean, not that, okay. All right, so will the tall and the short plant make a medium plant? Darwin set this up for years. Time to crossed hundreds of them. Are there medium plants? No, nothing. No mediums. 100% came out tall. Every single one of them. Not one of them was even short. How? That's not possible. We're supposed to blend. I don't know, Marvel hype might be dying soon. We'll see. Maybe not, maybe not. So, what he started to assume was that there must be one piece from mom, one piece from dad that informs the organism. What you're looking at today is the original basic version of Genesis, I'd say. A male gamete and a female gamete. Now, he didn't know what these were. He didn't know what they looked like. He's still under the assumption there's a piece of the mom, there's a piece of the dad that shows up. And those pieces combine into two. So if this is 1N, and this is 1N as we're calling it, as he's calling it, zygote, this new organism, all these tall plants that showed up in the breed, they must be two. They must be two parts. One part mom, one part dad. So equally, if he had stopped here, the picture would be quite incomplete. You would just say, oh, wow, well, looks like tall really gets the job done, I guess. One last thing that he did. If he bred all these tall children right here, these pea plants, he didn't get all talls. He didn't get clones or anything. This is where he gets everybody's favorite. He got 3 talls for every 1 short that showed up. So if he had done this 400 times, he'd have 300 tall plants and 100 short plants. Now, equally, what he had done, since technically all these tall plants would be the same genome, he did self-fertilize them. That's fine. So we end with this ratio here. If you had blended, and that's why this is up here, if you had blended, here's what the expected result was. You got a bunch of medium plants. If you breed the medium plants that all are the same height with the medium plants, they're all medium still. No blending can occur because they're the same thing. Instead, we got our good 100% offspring in what we call an F1 generation. And when we took these one more time, they produced 3 to 1. And this was the mystery for him. Now, Mendel, again, did pretty well with the math here. He started to build the ratios. He started to see things. But yeah, like I said, he was kind of a weird dude. So he did say, these are my bastard plants, the ones from the two parents. A little sad. So what we know now today, so we'll give some answers in here. We zoom in on these. We know that this tall phenotype then is a dominant trait. It shows up each generation. It sort of masks, and it hid that short one, but it did reemerge later, that short one. He termed the short one recessive because it can hide. It was masked 100% by the dominant phenotype. We're not going to spend too much time on the ratios yet. Here's the basic thing. And I know, like I said, I want to make sure this class hits at the molecular reasons for this. Okay. Going a little beyond what's here, what the ratios are, pretending that I can see inside the plant and know the moleculars. Okay. The gene that determines tall versus short has 2 versions of it. Everybody remember alleles? Good. Yeah. We're on the same page then. There is an allele, the dominant tall allele, that exists. That allele will make a gene that causes the plant stalk to be rigid and tall. Without that gene, that gene is sort of broken. It doesn't rise as high, kind of crumples up. That's a recessive allele. You will typically find that recessive alleles are simply non-working versions of genes. And that's why it only takes usually 1 dominant copy to get the job done for the dominant phenotype. That's the why behind that. Not quite there yet. It is day 1, and I know I'm kind of torturing everybody, but you're stuck in here for 30 more minutes, so let's keep going. Sorry. Okay. Major problem number 1 was solved for evolution. Heritability is not blending. They're discrete units. There's math behind it. They're discrete units being passed between mom and dad, and those ratios actually are on a dominant versus recessive sort of balance. Problem number 2 remained. Where do the weird ones come from? There's another French guy, De Vries. He was a botanist. He would go out and find this little type of weed right here in the fields that he existed near. He found that they were all the same plant, but they had really weird leaf change like variations to them. And remember, plants are a good organism because there are millions of these little weeds in a single field, right? Can't do this with like dogs, humans, other things that take a long time to have generations, right? In a single lifetime of studying, he collected something like 20,000 samples, and he defined that these are the same plant, just with variations. He called these mutants. These are Darwin's freaks that he needed. There needed to be some sort of source of randomness, of change. This plant was the first step towards showing organisms can have weird characteristics, and it can come suddenly. So, strangely, De Vries was the one that actually put it together in the end. He ended up finding Darwin. And in his search for plant science, he stumbled across Mendel's paper with the ratios, with the heritability, with the pea plants, and he saw it. He saw 3-to-1. He saw true breeding. He saw dominant recessive. And together, Wallace was sadly dead by this point. De Vries was the one that sort of said, okay, so this idea that was floated all this time ago, that there's variation in populations, we actually have the pieces now. Because if there's variation, there can exist some organism in that pile with an advantage. And in that, when that organism, and this is the important part, when that organism has an advantage, it doesn't just blend away, it doesn't just kind of dilute, it stays and it's strong. So this is a very big oversimplification, but good genes stay. It's day one. That was a big issue with, it's like, well, it might have a good gene for a while, but it's going to blend. Nope, they can stay. And then when the environment changes, when there's a drought, when there's a massive plague, when there's a food shortage, when food changes, when predators show up, that acts on this. And suddenly the game sort of changes. The economics of the population change. Okay. All right, let's see how this goes. So, fun times. No more notes for a bit. Let's try this. Poll everywhere, everybody's favorite. You should have seen during the Zoom year, because everybody's on their computers, like obviously not paying attention, but they'd be looking for a poll everywhere to show up, and they were just terrified of it by that time. They're like, oh my God, it's a poll everywhere. What are we gonna do? It was fine, but I'm glad that we're not in COVID anymore with online. Okay, so fun. If you didn't register, oh, shoot. At least we're here today. If you haven't registered, go use that time and like maybe try it now. Equally, you can today just come up with a sheet of paper and say, here was my response. I haven't been able to make it work yet, sorry. So, you know what we should do is feed these into, we should ask this question to the ChatGPT. I gotta be honest, that actually be kind of neat. It does not respond very well. Don't copy people's answers, stop it. So, it does not respond very well when you answer it and you give it an open question, I'd say. Think big though about genes, right? What about food? What about breeding? What about dogs, right? What about animals? What about medicine, right? Think about things that you've seen. Think about diseases. Think about immunology. All kinds of stuff. Think about CRISPR. Think about what it means philosophically to change the code. Think about what the history of genetics was, where it started. All kinds of stuff. Somebody last year put like fear of failing this class. And I was like, oh no, please, it's okay. We'll make it. It's okay. Yeah, so typically poll everywhere are a really good way for me to just kind of give everybody sort of a break, also see where everybody's at with stuff. This is typically where I'm going to do some practice type questions. This is also where I might do writings once in a while. They would only be worth 1 to 2 points because they're small. This is also where I'll do class feedback. I remember one time it was like, what was it? It was like, I was like, can't remember what the question was, but one of my students was like, pay me so that I don't have to go to school as much or something. Like that was his answer to one of our like topics. I was like, oh, dang it. It got so many upvotes. It like swarmed it. It was terrible. So yeah, if your poll everywhere is not going yet, crack a little piece of paper off before you leave. Go ahead and put it here and try and get that done before Thursday because we will start to ramp up how much we use this in some cases. Other times I'll actually have physically hand you pieces of scrap paper. My classes know to fear when they see the big pile of scratch paper showing up. Sorry. It's fun for you. Writing's good. You've been, you know, breaks up class a little bit. Equally, other online stuff. Do that survey. It's fun. You don't, and if you don't really want to do it, you can come talk to office hours with me and then that's it. That's easier. Equally, try that quiz, give it a shot. You have unlimited attempts. Do not stay at 80 or 90%. Just keep doing it until it's done, please. That's sort of just where we're at. That's always good for me to see. So those 2 things, main thing for this week. We'll see on Thursday. We'll kind of jump in with a small but targeted writing. We'll see. So fun times. I don't know if it's going to let me scroll. It totally is. Nice. Look at all this stuff. Yeah, proto-oncogenes. Yeah, somebody said cancer. There you go. Oh, did somebody say microbiome maybe? Yeah. Let's talk about changing science. That's going to be a big deal. You guys should take, micro with some of the upper guys that teach it. All right. Or yeah, you should definitely take med micro if you want to challenge. But everybody's like, it's hard. It's like, that's the point. Prepares you for med school. All right. Fun. Good job. So overall, I think that was like a decent chunk of you that have it working. So that's a fun time. Equally, if I don't hear from you though, I also kind of use this to try and find the people that forgot that they're registered in the class. Who's ever had that dream where you wake up and it's like, oh my God, it's week 8 and I've never been to this class. I'm going to fail. Just me? God, it's pretty terrifying. It literally still happens. It's the same English class in high school. It's like, you haven't been here all year. And I was like, oh shit, my mom's going to kill me. She was a teacher, so she would have. Sweet. There's one too. There's one that we kind of put together over the years. This one's a little scarier, sorry. I don't know what happened. It got a little more philosophical, a little immortality, fate, GMO, death, de-extinction. Soul and engineering are not meant to be next to each other. That just happened. Sorry. All right. Life, creation, future, disease, epigenetics, revolution, fun. So I wouldn't put this up if there weren't changes in the last 2 years, the last 10 years, obviously, that have changed things, especially from when I took this class. When I took this class, CRISPR was not a thing. A disease like double sickle cell was incurable and totally, that's it. There are patients now that have erased their sickle cell and gotten to be heterozygotes because of therapies we've made in the last 2 years. I watched a documentary on it, caught me by surprise. I was like, they did? Like, there's stuff like this that's happening that it seems behind the scenes. The goal of this class is to make that more accessible, though. And there's always a fun debate here to be had. First thing that really changed humanity was the atom. Once we blew up the first nuke, we knew things are different now. Once we got into a section or a power over information and informatics that the byte gives us, yeah, things have changed since then that are worse sometimes. The gene, the quote about the gene is that you control the gene, control the future. It's about food, it's about medicine, it's about a lot of things. It seems very dehumanizing sometimes too, but I do want to, we'll get into all the cool stuff and the hope and everything in that has in there. Okay, so, yeah, it's red, but we're bio majors. We know what a cell is, right? We're doing okay. I don't see anybody taking notes what a cell is. It's okay. But there are cells that exist alone as themselves. There are cells that form groups, that form multicellular complex organisms, like the cat apparently was the example here. The cells in each of us, although they form a unity to become something bigger, yeah, each cell is a cell individually. And as when we get into cancer, we'll see that when they start behaving like single cells, they turn into cancer sometimes. Now, each of these have a very core component of this class is that they all store information as DNA. DNA is the universal molecule for them. That's the recipe book. And they've all got it. Save for some red blood cells that ditch their genome because they're functional, and some of your immune cells, they mutate their own genome to get better. You don't need to know that for this phase of the class, though. Every functional cell with an asterisk has a full genome. Every one of your cells has access to every one of your genes, all of them, all 20,000. Philosophically, that means that your intestine cell has the same recipe book as your brain cells. It is, again, a question of wolf versus Pomeranian. What genes are you going to choose to be up and down, on and off? Everybody has every piece of info in those cells, though. Chemistry. That's why I put it in blue text. I don't want to freak you out. Yes. Do you need to know A goes to T and G goes to C? Yeah, I mean, that's going to help. And I kind of need that. In the second phase of the class, we get into some of the chemistry. But this is not a chemistry class. Thus, your expectations will be limited. Equally, who's read enough to the syllabus? What do you get to bring into the exam with you? Yeah, two-sided. Sweet. You get 2 sides of whatever you want. Do not have the hubris not to use that note sheet. Do not have the pride that you think you're going to make it. Because then with the note sheet, honest to God, with the note sheet, I can ask questions that actually mean something, right? I don't have to get us bogged down in stuff. If you can Wikipedia it, I don't want it on the test. That's my thing. The note sheet will help so much for both of us learn more, be better. That's it. Because again, if you can look up what does adenine look like, I'm not going to test you to draw it, okay? It's not worth it in this age. But we will confront it. But for now, it's just our nice substance that is the heritable material. It's what passes. Now, functionality-wise, every one of your genomes exists in every one of your cells, and it's a copy. There we go. So, in the genome, yeah, what are we looking at? I can't remember. It always fluctuates. 8 billion base pairs, 8 billion nucleotides. Nice. You got about 20 to 22,000 genes, as we call them. And that range differs depending on what you call a gene at this point. Most of us, most of us as in humans, 23 pairs, sorry. Wasn't going to say anybody doesn't. Depends on the animal or plant. This genome of 8 billion is divided into 23 pairs of discrete functional units, these chromosomes. We'll learn in this class why we make the discrete pairs and why it's not just a blob like prokaryotes and bacteria. And in here, in each of these chromosomes, you have genes, functional units that become something and have an effect on the cell, which then has an effect on the tissue, which has an effect maybe on the organism. So, again, you have the entire genome. That's like an entire book. You can look through the thing. And a book with 8 billion letters, it's still pretty big book. They printed the genome when they sequenced it the first time as like a show, and it was like stacks of paper. Now, if you want to zoom to a specific chapter, that's chromosome. Certain chromosomes do carry certain potentials. Chromosome 6 is one of your best sections of immunology genes, for example. You have an issue with chromosome 6, you have a bad immune system. But it's each gene, each sentence that tells a story. It can be something specific. I also like this example because when you change a sentence, the gene itself can change. So if you lose a few nucleotides from a small gene, I'm trying to think. You accidentally delete the information that would complete this sentence. Now it's a little scrambly, it doesn't mean as much. This one's not going to translate into anything. I'm going to bring in a sentence tomorrow that we can like play with and make it funny. But maybe you don't know it's a gray cat anymore. Maybe you lose pieces of that gene over time, and maybe that story would tell things better sometimes. But that's the basic hierarchy. I don't usually like doing rank questions on exams, but I mean, I would like you to know which one of these is bigger of the three. Oh, fun. All right, how's everybody's relationship with transcription and translation? This is our first try at this. Thumbs up, I feel okay. I feel pretty good. And you can do the close to the chest so you don't see it, so nobody sees that. Feeling okay. Yeah. And until we get into the second phase of the class, we don't have to approach it any different than right here. So you have DNA. You haven't run out of time, don't worry. You have DNA. Every single cell has this whole spool ready to go all the time. This is all here for us. Now, one advantage DNA has, and yeah, this is some chemistry, but I'll put it in blue here, is that it's stable. See how the nucleotides bind to each other? It's a good fit. Remember how it does a helix? It hides them. DNA is very hard to damage over time. You could leave a tube of DNA on a table, it'd probably be fine in like 2 weeks. No joke. So it is immensely powerful for storage. And it is immensely clean. It is your perfect vault from which to decide what you take from in each cell. And the chemistry helps this image, I think. But again, we're not quite like, I don't want you to get scared from that yet. Now, if you want to actually make something, if you want to make a gene for an immune receptor or defense receptor or a taste receptor, let's say, you do need to find that section of genes. I may as well just include the whole thing. So what happens is that when a piece of DNA is decided you will be activated, it will actually open up this helix and it will produce a second copy. So it would be like me saying like, all right, in the book it says the gray cat runs upstairs. I will take this over to a piece of paper and write the gray cat runs upstairs. That's the piece of paper is the mRNA. So this will be copied. You will not touch, damage, do anything to your DNA. It's safe. Because the DNA is in the nucleus, remember. Nucleus is a whole safe place, like a fortress. You don't want anything getting in there and mutating your DNA. Now, the RNA, it has a very short and rapid life. The cell is a very dirty place, sadly. A lot of chemicals, a lot of degraders, a lot of stuff. This mRNA will go out and it will exist for a brief moment. It is a copy of the nucleotide code. But RNAs functionally do not do 99% of stuff. Whenever there's something that's actually happening, like an enzyme chewing up something, or a receptor or a ligand reaching each other on a neuron, the stuff that actually does anything, it has to be protein. The mRNA's survival goal is to become and translate and produce a protein. It is the code that will be built, that will build that functional piece. I'll do this in red. So this is the functional thing. This is the real thing that you see on all our beautiful biology textbooks with the cartoons. They're the ones that are actually doing something. So in this exchange, it's not an exam, but think to yourself, and I know they're all important, but who's the most important? Would you rank choice these right here? So you can just think to yourself. And really the question might be easier, who's the least important here? Technically, if we had to say it. It's probably our guy right here. Sorry, middleman. You need DNA to store and pass on. You can't pass RNA on, it's too unstable. Remember what I talked about, how the helix forms and the nucleotides protect each other? RNA, nope. If you left RNA on a desk, it'd be done in 3 minutes, broken to pieces. Part of that is a facet. You don't want genes, you want to control gene doses. They can only have like a half-life of so long. As much as I love DNA too, you've got to have the functional protein. It has to work. It has to be there. It's the only way. So, another example that I would pose, it's a coding example, fun. I hate coding, but oh well. You never see the code of a website, but it's all there. Now, when you request something specific from a website, it will feed you that piece of code that will translate into something functional that you can click. You can scroll, you can see, you can comment, you can do whatever, right? The functional part is what we see. But in the inner workings of what it could come from and what it could be, it still exists in the background. Even though sometimes we're only requesting a small bit of it each time. Okay. So, yeah, it's in red text, but it's okay. I listed some of my best friends from my research on here just to show you that these genes have discrete locations on these chromosomes. Here, I'll scroll up here more. There you go. Now, not every organism has 23 pairs. We will encounter organisms that don't even have pairs. Sometimes something different can happen in certain organisms. But this gives you a rough estimate of the size. And you'll notice that, yeah, like there's some range on who counts stuff and where genomes count and how big they are and what does a repeated element count as. But the key here is that these genes all exist at certain spots. Oh, hideous typo. Oh, sorry. Well, not typo, but it like rolled off, basically. But each of these little genes, most of these are actually cancer genes that I study for the B cell cancer that I do. Oh, did it again. Sorry. They all have homes on these things. So, fun times for next lab. Anybody ever want to be a surgeon? Good times, your first patient. We're actually, the fly larva does sadly die from this, but you do actually get to extract a very DNA-rich section of cells from some fly larvae next week. Visualize the chromosomes as they spool out. And you actually get to see how big these things are, because usually they do, I mean, think about it, billions of nucleotides, they got to roll up pretty tight, but then when you actually heat them up and like squish them, they're huge, like super long. It'll be fun. So those discrete units that we talked about, they can be even more specific if this helps. Now, that's why I'm going to try, sometimes it'll feel like I'm repeating stuff, but I promise that it's with the intention that somebody may take a visual better than the original that I had. So some of you may do just fine with one piece mom, one piece dad. I'm a little odd that I like seeing everything. So this makes me a little happier here. But in this case, this is the full process here to create that zygote, that organism, and enjoy the fact that at one point, everybody in the room was just one of these. That's it. Kind of scary when you think about it, but hey. So, some verbiage again. Each one of those pieces, actually I should have noted it up here. Each one of these pieces is one N. Remember? with meiosis, like you saw in 105, the genome basically randomly splits into a bunch of combinations of chromosomes. This is advantage number one over the prokaryotes with their big blob of chromosomes. That's all they have. They just have a big blob. With us, you can mix and match. And as you combine in here, you reach 2N. Each one of us in here, 2N, 2 of each chromosome. We each have 2 chromosome 6s. We each have 2 chromosome 17s. Each one of those from a mom and a dad version. Now, this is where blending always kind of got its foothold, is we would look at human traits that are complicated to start. Or we would look at traits that are determined by a lot of genes to start, sadly. Height is one of those. Height is a very easy thing to look at. And typically, you're going to be about the average of your parents. So full genome-wise, yeah, you are about 50% of your parents. You are about 25% of each grandparent, thus, right? I'll say that's parent 1, there's parent 2. But we know that wholesale blending is not what's happening. You are just getting 2 discrete units from each one. You are getting a full complement from each one. Your mom gives you chromosome 1, 2, 3, 4, all the way to 23. Your dad gives you the same. They come together to form the pairs that make each of us. So, oddly philosophical, the chicken is technically the zygote's way of making the better zygote. Yeah, it's a bit of a little like brain circle. See if you can see where I'm coming from here. And trust me, I always still kind of am like, it's always a little much. But the chicken is the functional carrier of the information. And the information itself, though, strangely, philosophically, is infinite, right? The important genes in each one of us were important for all mammals, right? Let's say it's a protein that makes cells function, like actin or something fun. That information is what you can call sort of forever. And each of us, each organism is pretty much just the vessel for that information in a way or two. So, fun times. Like I said, genetics has opened so many fun but semi-dangerous doors in some ways. We'll talk about this on Thursday. We'll see the positive, we'll see everything else. I would say that the importance of this class is so many-fold, but one is that genomes are more accessible than they've ever been. Who's done 23andMe? Yeah. Yeah, they got us, don't they? Of course I was going to do it. I wanted to become a genetics professor. Of course I wanted that. But when something is accessible does not mean that it's being handed to somebody that has all the tools to understand things. I've had a lot of friends panic and be like, I have this gene, it says I'm going to die. And I'm like, oh God, please, please, please chill out. It's okay. Genes are risks. Genes are part of the equation. The massive information that you can sometimes give without interpreting it can lead into the dangerous proposition that we have right now, which is simple, easy answers to complicated problems are dangerous, okay? So part of this class is to be able to inform and figure out that gray area and discuss it. Equally, we're done. We finished early despite all of my stuff. So, fun times. Give me your little Pull Everywhere PE papers up here if you can't. If it didn't work, then go and try and register. Equally, try and do a little intro survey and the quiz so you get those free points. Fun. 5 points out of 500. That's a whole %. It's a good time.